briefly discussed about the differences between prokaryotic cell and eukaryotic cell. We discussed that eukaryotic cells, they have a true nucleus located in center, and the examples of eukaryotic and prokaryotic cells were animal cell, plant cell, fungi cell, and then we discussed about their composition in terms of their cell wall. A quick check from Abdul Rahman and Usman. Can you tell me that the cell wall of plant cell is made up of?
Cellulose.
Perfect. And what about fungi?
Chitin.
Chitin. Excellent. And okay, perfect, Hira. And what about bacteria?
Peptidoglycans.
Perfect. And what was the other name for peptidoglycan?
Murin.
Murin.
Yes.
Perfect. Excellent. So, okay, this is what we discussed briefly yesterday about prokaryotic cell and eukaryotic cell. Then we also talked about ribosomes, right? So we did not delve into the concept of ribosomes. We just discussed the differences between two of them. So we have two types of ribosomes. One of them is
Messenger RNA.
no, you have gone too far. You have stepped into chapter 5. I'm asking about the difference of shape.
Oh, 70 S and 80S.
Excellent. So yes, this is the difference of size between ribosomes. We have 70S ribosomes and we have 80S ribosomes, and they appear like dotted structures with two small subunits. We'll discuss it. We'll discuss it extensively in chapter 5 and in this chapter as well. So this was a quick recap of what we covered yesterday. Now moving on to the basic difference between animal cell and plant cell. So I believe you guys are already aware about the cell wall, that plant cells have cell wall made up of cellulose, but animal cells do not have any cell wall. Then plant cells have chloroplasts. Chloroplasts are the organelles responsible for the photosynthesis, and animal cells do not have chloroplasts. Then this is also one of the major differences between plant cells and animal cells, that animal cells have very small vacuoles and they are temporary in nature. So they get destroyed very easily. But plant cell has a very big centrally located vacuole. And we will also write one more difference, that the vacuole of plant cell, the membrane of vacuole in plant cell is known as tonoplast. Okay, so this is not mentioned over here, but this is also one of the differences, that plant cells have a membrane around their vacuole, and it is known as tonoplast. Okay, so this is one of the major differences between plant cell and animal cells, that animal cells do have vacuoles, but they are not surrounded with a membrane. Their membrane is not known as tonoplast. Then, so I believe these are two new terminologies that you guys are seeing, that plant cells do not have centrioles or microvilli, and animal cells have centrioles and microvilli. So we have these organelles in our syllabus. So we'll discuss extensively about the function, about the shape of centrioles and microvilli. So you do not need to worry about these at this point. Just keep this thing in mind, that these are also the differences of animal cell and plant cell. So far, are we good?
Yes.
Yes.
All right, perfect. Same. Okay. So now moving to the labeling of prokaryotic cell, we discussed it briefly. Can someone remind me that the hanging part of DNA is known as, there's one hanger for DNA.
Histone protein.
Histone proteins, perfect. So prokaryotic, do they have histone proteins? I'm talking about prokaryotic cells.
No, miss.
No, perfect. And what is plasmid?
It's
They
used in
contain
genetic
genetic
engineering.
engineering.
Yeah, perfect, excellent. And how are you going to differentiate between flagella and villi?
Miss, flagella is like long, and Pili has short
It is a
structure.
short-threaded structure.
Yeah.
And what is flagella responsible for?
For the movement.
The movement, excellent. And what about villi?
They provide like miss, I'm not able to recall.
Okay, कोई बात नहीं. So villi provide attachment to a eukaryotic cell. in order for bacteria to spread the infection from prokaryotic cell to a eukaryotic cell. Now moving towards light microscope and electron microscope. So we have two types of microscopes in our syllabus, and we need to do a lot of calculations for both of them, because your paper one will also have calculations, and your paper two will also have calculations regarding microscope. Okay, so we need to understand this concept of resolution and magnification extensively in this subtopic. So, are you guys aware about light microscope? Simple सा है ये तो. Have we seen a microscope? हमें पता है microscope क्या होता
Yes.
है.
And used in biology lab.
ठीक है. So it is used in biology lab, and why do we use it?
To see microscopic organelles of plant cells.
To see microscopic structures of any cell. Okay, so we have two categories, light microscope and electron microscope. And before we delve into light microscope and electron microscope, I want you to understand one concept of resolution and magnification, because these are the two terminologies that I'll be using while explaining light and electron microscope. So, ठीक है. So, we know that we have a nucleus in animal cell and plant cell, right? But we did not have this idea before that nucleus has two membranes. Okay, so we knew in O levels that nucleus is an organelle located in center, but we were not sure if it has two membranes or a single membrane. But so resolution is basically the ability that is differentiating between these 2 points, that nucleus has two membranes and not just one membrane. So basically what resolution is the ability to differentiate between 2 points. While in a light microscope, resolution is not very high. So this is why we are not able to see two membranes of nucleus. Rather, we just see one membrane of nucleus. So far, are you guys clear on this?
Yes.
Okay. So the resolution of light microscope is not very high, which results in light organelles not being seen in light microscope. For such as we can't see mitochondria having two membranes. We cannot see that chloroplast has two membranes, and we cannot see that nucleus has two membranes. So the distinguishing factor between 2 points is known as resolution, while magnification is the factor which enlarges the actual object, and we are able to see it with image size, right? So magnification is a enlarging property, while resolution is a differentiating factor between 2 points. Now moving back to electron and light microscope. So light microscope has a resolution of 200 nanometers, and you have to remember this resolution. You'll also have to remember magnification, and you'll also have to remember resolution of light microscope along with electron microscope. So light microscope has a resolution of 200 nanometers. Acha. So one example, that cell membrane has a size of 7 nanometers. Okay, cell membrane is of 7 nanometers. Can we see cell membrane with light microscope? Considering the fact that light microscope has a resolution of 200 nanometers, can we see 7 nanometers object?
No, miss.
No, exactly. So here comes the difference between electron microscope, that electron microscope can see organelles with their double membranes like nucleus or mitochondria or chloroplast, because it has a resolution of 0.5 nanometers. So with 0.5 nanometers of resolution, can we see cell membrane?
Yes.
Yes. So
Yes.
in light microscope, we cannot see an object which is below the size of 200 nanometers. So it is not having a very good resolution, right? Along with the magnification. So magnification is also just 1000x. X is the sign of magnification. You'll always have to add while writing magnification, that x10 or x20. And if you will not write x before writing the magnification, then you'll not get marks. Okay, so this is important. Then for electron microscope, we have magnification of x 250,000. So we can enlarge objects in electron microscope, and we can also see their detailing in electron microscope, because we can see extremely minute objects in via electron microscope. So far, are we clear? Okay. So you'll have to remember these pointers. Okay. Whatever I highlight during my lectures, these are important things that you'll have to remember. Because we'll be mentioning them into our scripts. And these are also the keywords that your marking scheme has already mentioned while answering such questions. Because there's a repeated question that what are the advantages of electron microscope over light microscope? So this is a very favorite question of CI. So you'll have to remember these points. Then light microscope can see living specimens. This is also important, which can be dyed. So we can only view living specimens in light microscope, and we can also put a dye on them. But for an electron microscope, our specimen needs to be dead. So we are only able to view a dead object in electron microscope, and electron microscope has a drawback that it can only produce black and white images and not an image with colors or with stains. Okay, so this is a disadvantage of electron microscope, that it can only produce white and black images, while light microscope can produce images with different stains. Then okay, so one more thing, that if we can see living specimens in light microscope, then we can also see many processes, like we can see a process of mitosis being happening, right? But in electron microscope, we cannot view different processes that are occurring within a living cell. So this is also an advantage of light microscope, that we can see systems, we can see processes happening live. But in electron microscope, we cannot see any live process happening in the living cell. Then light microscope shines light through the specimen. This light is then passed through an objective lens and an eyepiece lens, which magnify the specimen to give an image that can be seen by the naked eye. And since electron microscope, they have electrons, they have higher frequency of electronic waves as compared to visible light, the magnification and resolution of electron microscope is way better than light microscope. Are we clear so far? Have we understood the difference between light microscope and electron microscope?
Yes, ma'am.
And are we clear about, okay, Minha, do you have any question?
Yes, Miss, why can't we dye anything in electron microscope?
Because so electrons have a very high frequency, and if we put a living object under an electron microscope, it will automatically die because they have high frequencies, they have high radiations. So any living cell cannot bear those high frequencies or higher frequencies or higher wavelengths. It will automatically degenerate them. Okay.
Okay, thank you.
Abdul Rahman, do you have any question?
No, ma'am.
Okay. So moving on to electron microscope. Electron microscope has further two categories. It is a scanning electron microscope, and we have a transmission electron microscope. So scanning electron microscope, with name being said, we can already understand that scanning electron microscope can only scan the surface of the membrane. For example, see, this is a red blood cell, right? So we can only see the surface. We cannot see what is inside this cell, this red blood cell. While transmission electron microscope transmits the electron along the surface, and we can see internal details from transmission electron microscope. Here, do we have any question here? This is a very simple concept, but you have to remember this because they do ask you about the simple difference between scanning electron microscope and transmission electron microscope. You'll only see a 1 mark question regarding the differences between both of them. Yeah, Minha. Sure. All right, perfect. Okay. So now moving towards this important formula. This is highly important because you'll see questions of magnification, you'll see questions of image size, actual size again and again in your past papers. So magnification, if you want to calculate magnification, then you need to write image size in numerator and you need to write actual size in denominator. So they can give you two values, and they'll ask you to find out the third value. Image size is the value that we are going to find out from that we're going to find out from magnifying the object, while actual size, if, for example, this is an object, so the actual size is going to remain the same, but with the help of magnification, we are able to enlarge it, and the enlarged image is going to give us image size. Okay? And for magnification, what sign do we have? When we are finding out magnification, then what do we need to write before mentioning the value of magnification?
X.
X, perfect. ठीक है जी. Then these are very important conversions. You'll have to remember this. If someone is very bad at maths, then I can give you a small advice. Okay, so this is important. You'll have to remember this as well. So when we are going from centimeters to millimeters, then you multiply by 10. Then if you're going from millimeters to micrometers, you have to multiply by 1000. Then if you're going from UM to NM, then you have to again multiply with 1000. But if we want to find out a value in micrometers, and if the value is already given in nanometers, then what we're going to do, we are going to divide it. So change the color. So we multiply when we're going from cm to millimeters. We again multiply when we're going from millimeters to UM. UM is micrometers. Then we again multiply if we need a value in nanometers. So we multiply mm to UM in 1000. We also multiply UM to NM with 1000 again. But if we want our value in micrometers, and if the value given is in nanometers, then you divide it. Okay? So you'll have to divide 1000. Then again, you'll have to divide it with 1000. So to add division, I think I should use some other thing. Okay. Maybe then again, okay. So when we need a value from millimeters to centimeters, then we are going to divide it with 10. ठीक है? ये कन्वर्जन समझ आ रही है? क्या किया हमने? cm से millimeters जाएंगे, multiply by 10. mm से um जाएंगे, multiply by 1000. UM से NM जाएंगे, again multiply by 1000. फिर अगर रिवर्स में जाना है तो डिवाइड कर देंगे. क्लियर है? आ गया समझ में?
Yes, it's all clear.
All right. Perfect. ठीक है. Now coming towards organelles and functions. So we already mentioned yesterday that now we are not going to write cell membrane anymore in A levels. For the upcoming years of your life, now you need to mention that cell membrane is cell surface membrane or plasma membrane. You're not going to write cell membrane. You'll get a 0 if you write cell membrane. Okay, so cell surface membrane, let's talk about it. All cells are surrounded by a cell surface membrane, which controls the exchange of materials between the internal cell environment and the external environment. Wait. So, how is cell surface membrane able to control the exchange of materials? What property of cell surface membrane has enabled it to control the entry and exit?
Partially permeable.
Partially permeable, yes. So, cell surface membrane is partially permeable, which is allowing selective materials to move across the membrane. Okay, so this is a simpler thing. You already know it from your O levels. Then coming towards nucleus. So, nucleus is relatively large. Nucleus is relatively large and separated from the cytoplasm by a double membrane nuclear envelope. So now this is a new information, brand new information, that nucleus is surrounded with double membranes and not with a single membrane. What do we call the membrane of nucleus? We call it nuclear envelope. What was the name of the membrane of vacuole?
Tonoplast.
Tonoplast.
Excellent. So now the membrane of nucleus is known as nuclear envelope, and this nuclear envelope has many pores, and we'll discuss why it has pores in, I believe, in chapter 5. It has an important function. So we'll discuss this in chapter 5, why it has many pores. But right now you need to remember that nucleus is a double membrane organelle, and the name of that membrane is nuclear envelope. Okay, so now moving towards what is within the nucleus. So nucleus contains chromatin. Chromatin are thread-like structures which contain genetic information, and the condensed form of chromatin is known as chromosomes. All right, so you need to remember the difference between chromatin and chromosomes. Chromatins are uncondensed. They're thin thread-like structures, and both of them contain genetic information. While when the cell is dividing, those chromatin becomes chromosomes. They become highly condensed, and nucleus also contains histone proteins. Histone proteins are only present in which type of cell?
Eukaryote.
Eukaryotic. And at least one or more darkly stained regions can be termed as nucleolus. So nucleolus are responsible for producing ribosomes. We talked about ribosomes, right? That there are two types of ribosomes. So nucleolus is a spherical body within the nucleus, and it is responsible for producing ribosomes. ठीक है? Clear है? I'll ask questions. Then we'll come to know if we are clear or not. All right. So now moving towards this structure of cell membrane, and you do not need to worry about this structure because we already have a separate chapter for cell membrane. We'll start with cell membranes chapter right after we complete this chapter of cells. So we'll discuss about this structure. So this is exactly how your cell membrane looks like. It's not like this, like we used to draw in O levels. It is something like this, and it is a very complex structure, and we'll study extensively about it in chapter 4. And we'll begin chapter 4 right after completing cells because it's a simpler chapter. Then, okay, so let's discuss about the structure of nucleus, and then we'll take a break. We'll take a namaz break. So coming towards nucleus, we know that nucleus is located in the central position of animal cell, and it is located towards periphery in plant cells. Periphery means it is located towards the sides because in center a large vacuole is present, right? So nucleus has a double membrane known as nuclear envelope. Here it is written. This is nuclear envelope, and you can see these pores. These are the pores which nuclear envelope has. Then in the center, we have nucleolus. This is the spherical body, dense spherical body. We can stain it extensively, and this is responsible for the production of ribosomes. And then what else do we have? Yes, we also have thin thread-like structures. They are known as chromatin. And when the cell has to divide, these chromatins become something like this, and we call them chromosomes. ठीक है? Clear? So this is also a nucleus, right? And this nucleus has a spherical body. This spherical body within the nucleus is known as nucleolus. Then the cytoplasm of nucleus is also known as nucleoplasm. You will not call it cytoplasm anymore when we are talking about nucleus. Inside the nucleus, the cytoplasm will be known as nucleoplasm. And these are the thin thread-like structures known as chromatin. It contains genetic information, and they also become condensed when the cell has to divide. Then you can see these double membranes, right? And these double membranes are known as nuclear envelope. Between them are small pores. This is a small pore. This is also the small pore. This is also the small pore. So nucleus has nuclear envelope, which has many nuclear pores. So far, are we clear? Minha, you raised your hand.
Miss, I'm a little confused in inherited protein.
In which protein?
I don't remember the name, but it was like heratin,
Histone.
histone.
Yeah. Okay. So we discussed histone proteins yesterday. Wait, let me tell you. Abdul Rahman, you can unmute yourself. Let me know what is your question. So, okay, histone proteins are basically the hangers of DNA. Your DNA looks something like this, right? So we have two types of DNA. One of them is linear DNA, other one is circular DNA. So linear DNA, this is a linear DNA, and this is present in animal cell. While a circular DNA, something like this, is present in your prokaryotic cell. See, something like this. So this is a prokaryotic cell, this is a bacterial cell, and it has a circular DNA. So why is it circular? Why is it not linear? It is because of histone proteins. I mentioned yesterday that histone proteins are like the hanger of DNA. If histone proteins are present, then your DNA is going to look like this because it is hanged with histone proteins. But if histone proteins are not present, then your DNA, not your DNA, but bacterial cells' DNA will appear like this because it does not have histone proteins. Make sense?